In the striped or voluntary muscle, the nerves supplying the muscular fibers are derived from the cerebrospinal nerves and are composed mainly of medullated fibers. The nerve, after entering the sheath of the muscle, breaks up into fibers or bundles of fibers which form plexuses and gradually divide until, as a rule, a single nerve fiber enters a single muscular fiber. Sometimes, however, if the muscular fiber be long, more than one nerve fiber enters it. Within the muscular fiber, the nerve terminates in a special expansor called by Kuhn, who first accurately described it as a motor end plate. The nerve fiber, on approaching the muscular fiber, suddenly loses its medullary sheath. The neurolemma becomes continuous with the sarcolemma of the muscle, and only the axis cylinder enters the muscular fiber. There, it at once spreads out, ramifying like the roots of a tree immediately beneath the sarcolemma, and becomes embedded in a layer of granular matter, containing a number of clear oblong nuclei, the whole constituting an end plate, from which the contractile wave of the muscular fiber is said to start. Ganglia are small aggregations of nerve cells. They are found on the posterior roots of the spinal nerve, on the sensory roots of the trigeminal, facial, glossopharyngeal, and vagus nerves, and on the acoustic nerves. They are also found in connection with the sympathetic nerves. On section, they are seen to consist of a reddish-gray substance traversed by numerous white fibers. They vary considerably in form and size. The largest are found in the cavity of the abdomen, the smallest, not visible to the naked eye, exist in considerable numbers upon the nerves distributed to the different viscera. Each ganglion is invested by a smooth and firm, closely adhering, membranous envelope consisting of dense areolar tissue. This sheath is continuous with the perineurium of the nerves and sends numerous processes into the interior to support the blood vessels supplying the substance of the ganglion. In structure, all ganglia are essentially similar, consisting of the same structural elements, that is, nerve cells and nerve fibers. Each nerve cell has a nucleated sheath which is continuous with the neurolemma of the nerve fiber with which the cell is connected. The nerve cells in the ganglia of the spinal nerves are piriform in shape and have each a single process. A short distance from the cell, and while still within the ganglion, this process divides in a T-shaped manner, one limb of the crossbar turning into the medulla spinalis, the other limb passing outward to the periphery. In the sympathetic ganglia, the nerve cells are multipolar and each has one axis cylinder process and several dendrons. The axon emerges from the ganglion as a non-medullated nerve fiber. Similar cells are found in the ganglia connected with the trigeminal nerve, and these ganglia are therefore regarded as the cranial portions of the sympathetic system. The sympathetic nervous system includes those portions of the nervous mechanism in which a medullated nerve fiber from the central nervous system passes to a ganglion, sympathetic or peripheral, from which fibers, usually non-medullated, are distributed to such structures, for instance blood vessels, as are not under voluntary control. The spinal and sympathetic ganglia differ somewhat in the size and disposition of the cells and in the number of nerve fibers entering and leaving them. In the spinal ganglia, the nerve cells are much larger and for the most part collected in groups near the periphery, while the fibers, which are mostly medullated, traverse the central portion of the ganglion. Whereas in the sympathetic ganglia, the cells are smaller and distributed in irregular groups throughout the whole ganglion. The fibers are also irregularly scattered. Some of the entering ones are medullated, while many of the leaving the ganglia are non-medullated. Neuron theory. The nerve cell and its processes collectively constitute what is termed a neuron. And Waldeyer formulated the theory that the nervous system is built up of numerous neurons, quote, anatomically and genetically independent of one another, end quote. According to this theory, the neuron theory, the processes of one neuron only come into contact and are never in direct continuity with those of other neurons, while impulses are transmitted from one nerve cell to another through these points of contact, the synapses. The synapse or synaptic membranes seems to allow nervous impulses to pass in one direction only, namely from the terminals of the axis cylinder to the dendrons. This theory is based on the following facts. That is, 1. Embryonic nerve cells, or neuroblasts, are entirely distinct from one another. 2. When nervous tissues are stained by the Golgi method, no continuity is seen even between neighboring neurons. And 3. When degenerative changes occur in nervous tissue, either as the result of disease or experiment, they never spread from one neuron to another, but are limited to the individual neurons or groups of neurons primarily affected. It must, however, be added that within the past few years, the validity of the neuron theory had been called into question by certain eminent histologists, who maintain that by the employment of a more delicate histologic method, minute fibrils can be followed from one nerve cell to another. Their existence, however, in the living is open to question. Mott and Marinesco made careful examination of living cells using even the ultramicroscope, and agree that neither Nissl bodies nor neurofibrils are present in the living state. For the present, we may look upon the neurons as the units or structural elements of the nervous system. All the neurons are present at birth, which are present in the adult. Their division ceases before birth. They are not all functionally active at birth, but gradually assume functional activity. There is no indication of any regeneration after the destruction of the cell body of any individual neurons. Fasciculi tracts, or fiber systems, are groups of axons having homologous origin and homologous distribution, as regards their collaterals, subdivisions, and terminals, and are often named in accordance with their origin and termination, the name of the nucleus or the location of the cell body from which the axon or fiber arises, preceding that of the nucleus or location of its termination. A given topographical area seldom represents a pure tract, as in most cases fibers of different systems are mixed. End of section 2.